My name is Jacob Henson, and I chose to discuss prompt 2.2 as it relates to my eukaryotic organism being a sheep. And so here we have our three different loci that are being carried on these two alleles, and I'll be discussing one that carries a disease, one that carries a particular structural issue, and another that relates to muscling in sheep. And so that disease that's being carried is going to be whether they are scrapie resistant or scrapie susceptible. And then whenever we talk about structure, we'll be talking about whether they are dwarfism or having dwarfism, or whether they are normal structured animals. And when we talk about muscling, I'll be talking about whether they carry the callipygian gene or they are non-callipygian. And so scrapie is a disease that affects the nervous system of animals. Dwarfism, obviously, as you can see in the picture in the middle, affects structure. And so they're going to be smaller animals, smaller framed, bigger muscled animals, but they clearly have structural issues happening. And then callipygian relates to the muscling in an animal, and hypertrophy in the sheep. And so here we have our genetic 3-point map, and we have our dominant alleles on the top of that 3-point map and our recessive alleles on the bottom of that map. In region 1, we have a 19.28 centimorgan span, and in region 2, we have a 14.76 centimorgan span. And so any particular non-crossover event will result in 0.6596, that probability of that happening, and here are the calculations for that. And in any double crossover event, we will have a 0.0071 probability of any type of double crossover event occurring, and here are the calculations for that. And here is a table of phenotypes with a coefficient of coincidence of 0.573, and I tested a total of 1,348 progenies. And on the right, we have the probability of any of these specific events occurring here. And here we show the chromosomes. And so on the left, we have just a basic chromosome combination, that there are no crossover events on this left side. And so here I've kind of shown you like the different regions, different colors, and hopefully you're able to see that. And so in a non-crossover event, there is no exchange of these particular regions. The chromosomes will stay in place. And so here you'll see the different crossovers in these different regions. And so in a double crossover event, you'll be able to observe these particular regions crossing over, obviously. And so here we have our links for reference.